Professor Irene, welcome to the show.
Thank you.
I'll just take the opportunity and a little bit talk about your background. So, Professor Irene Ghobrial is the SVP for Experimental Medicine and Director of Center of Early Detection and Interception of Blood Cancers at the Dana-Farber Cancer Institute. There is a say said by Dr. Ghobrial, which is, we don't watch and wait, we shift it to early detection and intervention, which we'll speak about it
Yeah,
later on.
absolutely.
So, welcome to the show.
Thank you.
And to begin, just could you please take a moment to introduce yourself and your role at your prospective organization?
Yeah, absolutely. So I'm a professor of medicine at Dana-Farber Cancer Institute, Harvard Medical School. I've been there for 20 years, but you probably can tell from my accent. I'm from Egypt originally. بتكلم عربي and
I didn't.
excited to be here, excited to be part of an amazing early, development of collaborations between our institutes.
Thank you very much. So, let's shift gear now. And the first question is, what is the primary focus of your work within the healthcare industry?
So I'm an oncologist, and for many years, when we see patients, and the disease that I see is called multiple myeloma. It's a
Right.
blood cancer, and for many years, we see patients when they have symptoms. They get diagnosed by their primary care doctor because they have symptoms. They have anemia. They have fractures in their bones. They have pain. They come and see us, and then they're diagnosed with cancer. For the last 10 years, I started to say, well, that doesn't make sense. We wait until the cancer develops. And in many cases, it's already stage 4 cancer. It's already too late to see them. And then you diagnose them and then you treat them. What if we diagnose cancer early? And instead of waiting for it to happen, you actually find it and detect it early, because when you detect it early, you can potentially prevent it or intercept it and make sure that we don't have the late stages of the disease.
Right. So, it's more of a screening method, I would say?
So it's not just screening, because screening for certain cancers, it's fine. If they are solid cancers, you can screen and detect and potentially cut the cancer out. But this is more about cancer detection early and interception. For blood cancers, you can do that in a blood test and you can find it early. And then the question is this truly a precancerous lesion that will occur and cause cancer in the next 2 years, in the next 5 years, or in the next 50 years?
Right.
And you have to really risk stratify your patients, and then you have to decide on which therapy you can give them at that time.
Right. Simply, I could say that your research on multiple myeloma and its precursor condition is truly groundbreaking. For the benefit of our audience, could you explain what monoclonal gammopathy of undetermined significance, MGUS, and smoldering multiple myeloma are, and why early detection is so critical?
Yeah, absolutely. And it's a big, long name, MGUS. So, multiple myeloma is a disease that we found, at least in the US, in about 35,000 cases. But every single patient who has multiple myeloma must have had a precursor condition for years and years before.
Yeah.
And if you think about MGUS, or monoclonal gammopathy of undetermined significance, which is that long name, it's actually very common. 5% of the population in the US over the age of 50 has MGUS. And probably, we don't know very well, but probably in the Middle East, many of us have those precursor conditions.
Right.
So we all walk around with precursor conditions, MGUS or CHIP or MBL, which are the precursors of blood cancers as we get older, and yet we never get diagnosed with them until we have symptoms. Here you have an opportunity with a blood test to diagnose it early, to know when the patient will develop the cancer, whether it's myeloma or leukemia or lymphoma, and to potentially prevent it or intercept it early.
Right. So back to your say, which is
Let's not
shifted
watch and wait. Yeah.
let's not watch and wait, but instead detect and intervene.
Yes, absolutely. So the question of detect and intervene matters a lot. We started asking the question first, let's diagnose the patients correctly and define who will develop that. So in my lab, we do a lot of whole genome sequencing, single cell sequencing, to really identify who is the patient who will benefit from therapy early and what kind of therapy. And then for the interception, or for the treatment, we started shifting towards immunotherapy, which is either bispecific antibodies or CAR T therapy. And we actually tested it in our clinical trials for interception in smoldering multiple myeloma. And we have impressive data to show that they have deep remissions, what we call MRD negative, or minimal residual disease negative. And potentially in the future, we will get to know whether they're cured or not, but at least it's a curative intent to treat early. And hopefully you never get myeloma in your lifetime.
That's actually really interesting and fascinating. So, but I got a question, is that how do we know whom to, let's say, screen or to do a blood test for?
Yeah, great question. So should you screen the whole population, or should you screen people who are at risk? Right now, at least in the US, we're screening people at risk. So we are screening 30,000 individuals who are either of African descent or African Americans, or people who have a family history. But that's because myeloma is more common in those populations,
Right.
or as we get older. Now you can start thinking of different populations, different screening, and truly that should be the personalized way of screening rather than screening everyone.
Mm.
Currently, we screen only for four cancers. We screen for breast cancer, lung cancer, and even lung cancer is not screened correctly all the time. Colon cancer, you can argue about prostate cancer and cervical cancer.
Right.
And even with those, we're missing so many of those patients who are not getting screened, but
Mm.
we're also missing a lot of opportunity for finding pancreatic cancer or other cancers, and of course, blood cancers, because they're easy to detect in a blood sample
Mm.
and you can do something about them to prevent them.
And could you see that in the future? Probably we're going to screen for all of them.
Potentially. I mean, there are so many studies now that are using circulating tumor DNA, circulating tumor cells that you heard about earlier today, as well as many other methods like methylation and so on to detect cancers early. It all depends on sensitivity and specificity. And you may find a battery of tests that would work for different cancers at different sensitivity and different tests.
So this is really important. Early detections mean a better survival.
Absolutely. If you find it early, you detect it early, you can have a huge survival difference.
Yeah, and if we compare it, I mean, if we go back and what we've done through colon cancer, what we've done in breast cancer as well, it's a groundbreaking. This can be really great if we could just copy that to blood disorders, blood cancers.
Yeah. Can you think about if you have a relative who has leukemia or myeloma
Right,
or lymphoma,
right,
can I detect that early and prevent it
right.
completely?
So, what are the most promising areas of research and development in the field of oncology today?
Lots of great things. I think we're always excited. That's the best part. Every day is a new technology and a new development. I think right now we're doing a lot of spatial genomics and spatial transcriptomics. So instead of doing single cell sequencing that we usually do on fluid, we're trying to actually look at the location and localization of the immune cells and how close they are to the tumor cells. And that interaction and localization can tell you whether those cells will actually be able to predict response or resistance to therapy or future progression or
Right.
no progression. I think AI is infiltrating everything, big data, of course. So lots of opportunities for us to think of those multi-omics and then taking it into the future with AI to understand better and develop better biomarkers, but also to develop better precision therapy for our patients.
What about genomic sequencing?
Absolutely. I mean, DNA sequencing is the basics that we do all the time.
Right.
So, whole genome sequencing, taking it at the single cell level. But I think thinking of transcriptomics and proteomics is the future to really capture that whole understanding of the multi-omics of a cell
Right.
and then understanding how to use it for therapeutics.
Like genomic sequencing could help you determine which medication, which sort of treatment is better for this patient, not that patient, even though they do have the same disease, right?
Absolutely. So there are different things that you can think of genomic sequencing. You can think of germline sequencing, which is the DNA from your parents. That's critical for potentially response to certain therapies because you may respond or not based on some of those, or you may have different PKs of the drug. And then there is, of course, the genomics of the tumor cells, which is what we call somatic mutations. And this then can help you with those precision therapies. If you have a RAS mutation, you may respond to a RAS inhibitor or drug specific for that. And this is where precision therapy matters. And I think you need both to understand risk of the drug itself and how you're going to metabolize it and whether you respond to it or not based on your both somatic as well as germline sequencing.
Right. Going back to don't wait, don't watch, detect and intervene. I'm sure you had challenges shifting from one approach to another one.
Oh, absolutely. I think a lot of people still think that it's crazy to do that. And I would say,
Right.
again, everything that you think about, you have to really change the way we think and you have to innovate. The first time we thought of doing CAR T therapy, which is used really in heavily pretreated relapsed refractory setting, we said, no, we're going to take it into the smoldering setting, which is before the patients are diagnosed. And when we submitted it to the FDA, of course, the FDA said, absolutely no way. But they're the ones who actually said, yes, let's start there because the T cells are much more functional early on, and you can potentially induce a really deep remission with those functionally less exhausted T cells if you're using it earlier when it's less genomically complex tumor cells, and you could potentially have a curative intent. And indeed, we tried that and we're very successful so far with many of our treatments.
That's awesome. I think probably in the future, those numbers of cancers is going to be reduced, and we're going to be speaking about it as if we're speaking about like measles, like long time ago.
Yeah, vaccines. Think of it more like cardiovascular risk, right? Right now, you go to your primary care doctor, you check your blood pressure, you check your cholesterol. They'll never say, well, wait until you have the heart attack and then I treat you. This is how we need to think of cancer. Don't wait until you have the cancer and then I treat you. Think of prevention, think of finding the biomarker for that, which is the cholesterol, the blood pressure, and preventing it by taking something to make sure you never develop cancer.
Right.
And hopefully, in our lifetime, none of us will develop the cancer. We can detect it early. We can identify who will develop cancer in their lifetime, but we can potentially cure it early.
Inshallah. It's like doing your risk assessment and then start intervening directly.
Correct.
So you're here at Sharjah next healthcare event and the rapid growth in biotechnology ecosystem in the region. What is your impression?
It's so exciting. My sister lives in Dubai, and I came to Sharjah today, and I was just so amazed by how much innovation and technology developments you have here. It's really mind blowing. I always think like, oh, we are the innovators in Boston with the MITs and the Harvard and everything, but truly impressive to see how fast people think of developing something, but making sure that it happens here. So excited to see potential collaborations.
Perfect. Thanks. And could you share some of the insights into your innovative work at your lab, which are you conducting right now?
Lots of things. I think the best part about innovation in the lab is the postdocs always come up with innovative ideas, and it's the exciting thing because you have international students and postdocs from all over the world. We're thinking mainly of how to think of truly intercepting and developing new therapies in the next 5-10 years. Can we take all of the single cell sequencing we're doing and identify potentially T cell receptors that are specific for the tumor cells and then developing therapies from there? Or can we identify novel proteins on the surface of the cancer cells that can be the next generation of a CAR T or a bispecific antibody? Or can I develop a model based on AI that takes all of the multi-omic information that we have, proteomics, metabolomics, single cell sequencing, immune cell sequencing, as well as the clinical data of a patient, and put it together so that when you see a patient, you can identify in a more precise way who will develop cancer and who will not.
And start treating them.
And then start treating them, absolutely.
Thanks a lot, Professor Irene. It was really fascinating. It's an eye-opening. It's an eye-opener not only for me, for everybody, actually. We need to know more about cancers and start treating them, not wait and watch. No, detect and intervene. Thank you very
Absolutely.
much, Professor
Thanks
Irene.
again.
Appreciate it.
Absolutely.